The vaccine against coronavirus is the first RNA vaccine approved for human use. So the vaccine contains RNA instructions for the coronavirus spike protein wrapped in a lipid nanoparticle. One of the problems is that this vaccine needs to be cold, sometimes at sub-freezer temperatures. Why is this? The main reason is that RNA is less stable than DNA for many reasons. DNA has a double helical structure, which gives it stability. Much of the DNA is wrapped around histone proteins, which adds stability. The nucleus contains enzymes which repair DNA through multiple mechanisms. There are enzymes which can degrade both DNA and RNA, DNases and RNases. RNases are far more prevalent. The sequence of DNA is more stable for a couple of reasons, but that doesn't directly impact the vaccine. Thymine offers more protection from photochemical reactions, but a main reason relevant to the vaccine is that ribose is fundamentally more reactive than deoxyribose. As I review more fully in other videos, DNA and RNA are long chains, polymers, which are made of building blocks known as monomers. Each of the nucleotide monomers is made of three separate components: a nitrogenous base, a phosphate, and a pentose sugar, either ribose, which has an oxygen attached to its number 2 carbon, and deoxyribose, which does not have an oxygen attached to its number 2 carbon. The problem with that OH group on the number 2 carbon of ribose is that the phosphate group can react with it inappropriately. If a phosphate reacts with the OH group on the number 2 carbon, it breaks the RNA chain. If a batch of RNA vaccine was not kept cold, like the batch on the right, then there would be more reactions between the number 2 carbons and the phosphate groups, which would break RNA chains, and there would be a greater risk of degradation by the RNase enzymes, which are fairly ubiquitous. If the first 2 vaccines against coronavirus were both RNA vaccines, why would one require a lower refrigeration temperature than another? Well, it could be that differences in the RNA sequences of these vaccine RNA molecules could result in differences in the folding of the RNA, which would affect stability. There could be differences in the composition of the lipid nanoparticles, which help stabilize the RNA. And it may be that we may never fully know the answer because some of this information is maintained secret as being proprietary and confidential for those who are manufacturing the vaccines.